Okay, good morning, everybody. Welcome to the 11th talk in our series. It's going to be protein crystallization with Fabrice. But first of all, just like to say that we've got speakers then confirmed for the bioinformatics and for the hydrogen material exchange later on with Tim Stevens and Mark Shale. So let's go back to today, and I'll stop sharing my screen. And then Fabrice, if you want to share your screen.
Hello, everyone. Hey, I'm Fabrice Gorrec. I'm responsible for the crystallization facility at the LMB since 2007. The facility is open mainly to the scientists at the LMB, where notably users from the Department of the Structural Studies, PNSE, that are the main users. It's also open to people of the university, Cambridge University wings, called Immunology wings now. On this presentation slide, the first one, the picture is representing the miniaturization of the crystallization experiments over few decades. On the left, you see a flask containing some vials from experiments of Max Perutz from, I think, this date from the 70s. This is just to show that, matter of scale, the vials contain few milliliters of precipitant. I think it's phosphate precipitants and some hemoglobin. And next to it, you have a plate which represent the beginning of the pipetting, basically, mostly manual. I think this plate was in late 80s, and you can see now we proceed with drops. And they are pipetted in the plate. Now the scale is a few microliters per experiment. And then the third place is a now called SBS format plates, and then it represents the approach, the automated approach. Now the scale is 100 nanoliter drops, and on the far right is your macrofluidic chip, where now the experiment scale is in order of 1 nanoliter. So this was just a little overview. We're going to go back to miniaturization of experiments later, but I like this picture. This represents an evolution of the size of the experiments through time. Right, so I'm going to try to introduce macromolecular crystallization. We talk about macromolecular crystallization, which is applied to other things than proteins, protein complex with DNA, RNA. So that the approach is employed for different types of macromolecules, but we usually stick to say with protein crystallization usually. So there are a lot of aspects that could be talked about, and I will try to introduce a multitude of them, not all of them. So I will try just to place back the main context of X-ray crystallography and the main aspect of it, just to make sure we are on the same page of why this is for. I will try to see the characteristic of the macromolecular crystal, which unfortunately are quite unfavorable in most cases. The main aspect is the sample to be successful in crystallization experiments, and that I will cover also a few aspects there. Then we'll see the crystallization experiments, not all aspects. One main aspect is the solubility, and I will just show the notion of phase diagram and also explain the vapor diffusion experiments, which is the most widespread technique to crystallize protein. I will unfortunately skip with the nucleation and growth part because it was getting a bit too much and it was getting too long. So that will be for another time, unfortunately. Also, it's very interesting, but something had to go, and this part will go today, which lead me to initial screening in the vapor diffusion nanoliter droplets, because that's how we proceed nowadays in nanoliter droplets. We're not anymore at the scale of milliliter. All of this, of course, because the sample amount is often limiting. And I'm going to go through our main approach at the LMB, and including our strategy, which is a large initial strain. Then I will go and see some aspects of the assessments of your droplets now. After a few hours and few days, you will have a look at them under the microscope and see what you've got. If you got lucky, you can see nice crystals. And I will also briefly show some optimization protocols. The most common one, there are 3 that we are using at the LMB. There are more, but there are some that you must do to have a chance to optimize your crystals. And we'll finish with a few experimental tips. So just quickly, we're talking here about X-ray crystallography to determine a crystal structure. So the first main step is about producing some protein, and then you start with molecular cloning and expression. Here there's some, and put on the right the term ingenuity, because that's where, you know, it's a bit the smartest part maybe of the process, where you have to really think through a lot of things at the molecular biology level to have a sample that both makes sense for the study and have a chance to crystallize. Another important step, which is very time consuming, is the protein purification. And just to mention that here you need at the end to have sample of with few milligram per mil to have a chance for it to crystallize. So that can represent a large amount for some projects. It depends how hard it is to express and concentrate. Then we, of course, once you have this sample, you will try to crystallize it. And the main technique is vapor diffusion. Here, I mentioned that it's unpredictable. So the unpredictable aspect is why we are using a lot of different conditions to attempt crystallization. Then you need to, of course, what you want is some data from your crystals. And for that, you will submit to X-ray diffraction experiment. And I mentioned that you need to have at least 3.5 angstrom diffraction. This is when the data start to first to make sense, and when you have a chance to be able to process it, to have some interesting data. The structure determination now, most people will ship their crystal to the synchrotron, which is lots more powerful and which is tunable X-ray beam. And in the background, the IT system now means that you can solve a structure, if you have the right data, of course, and a very fast way. You can screen the crystal faster and you can solve the structure lots faster now. Just to mention diffraction aspects here, so that the crystal is a periodic arrangement, 3-dimensional arrangements of macromolecules. In that sense, it's an amplifier of the weak X-ray scattered by the crystal, as long as the conditions of Bragg laws are respected. If your crystal is big enough and the internal quality is good enough, then you will see a diffraction pattern that you record the diffraction pattern. And what you do, you want a 3-dimensional structure, so you turn the crystals, you rotate it, and you collect data in several angles to get different, to have the data necessary to solve the structure in 3-dimensional structures. Unfortunately, to calculate the electron density, you don't only need the intensity of your diffraction, the reflection. You also need phase angles, which you can't, it's not recorded, so you need to calculate that. And it's often a limiting step at that stage because the proteins molecules are relatively big and the crystals is far to be perfect. in general. So historically, an experimental method to deduce the phase is isomorphous replacement. So this is the initial protocol developed by Max Perutz in the 60s. Why I'm mentioning this approach is because you will need crystallization, some crystallization efforts to do that, because you need to obtain the exact same crystals as previously, but with a heavy atom, with a metal integrated into it after soaking. And from then you may have the way to deduce the data that is based only in the heavy atom location. And that's allow you to deduce the phase data. There are other ways to obtain the phase, like a molecular replacement, of course. But I mentioned isomorphous replacement because this is about crystallization. So here, this picture is a historical one. This is a, the name is Strandberg and Dickerson. Sorry. And they hold, it's outside what was becoming the LMB. And what they hold is stripes with data that are going to be processed by the very early computing system. So it was a struggle, obviously, all those calculations before. So that's just to mention that they don't have the same bottleneck as now, which is crystallization. They use readily available some proteins, like I think the first protein structure was with myoglobin of sperm whale. And it crystallized relatively easily as well. So they had all the problems in crystallization at that time. And it evolved with the structure of hemoglobin in the 60s and 70s with the work of Perutz and Kenju. And here is the flask again that we saw at the very beginning. It's here back because when you see the diffraction pattern, it's to say that this crystal still diffracts today. Well, we didn't check recently, but Jan and his checked that I think 10 years ago, and it still diffracts very well. So they're very stable as well. So stability of crystal were not such an issue before. And we didn't have nice picture on the computer with a three-dimensional structure, but there was some model made with wooden parts and wires. I think this one was made by Rostock at the LMB, actually. So the current context is different. So data processing and structure solution is done within hours on a laptop. So we don't have the same problems than in the past. But the more we solve structure, the more we work on protein sample, the more we end up with some that are difficult to crystallize. So the nature of the sample are increasingly challenging. So now the crystallization is really the main bottleneck, even more than ever. So we have a low yield of novel useful crystals for sure. And also in the background now, there's the competitiveness of cryo-EM, which is very attractive, obviously, to solve very large protein complexes. So that's a data collected from the LMB users. It's a bit outdated now, but it will be roughly the similar type of yield we'll have today. And basically, these are a lot of different type of samples, and I ask what's the yield once they obtain some useful crystals. And I calculated an average of 1%, as you did over when you start, you see the initial condition, you have more than 1,500, more than 1,600, up to 1,820 conditions. And it was just to represent the bottleneck of which is obtaining useful crystals. And that's only when some useful crystals were obtained, actually. So there are often optimization of the sample required before that where you don't obtain crystals. So overall, the yield is actually less than 1%. Now, here is a bit more fresh data, which is over the last 3 years. And I look at the deposition in the PDB. structures. So it's with the X-ray method compared to the cryo-EM method. And as you can see, the number of overall structure for the X-ray over time is still so solid. So there's not less structure solved. So still a lot of effort put into that. Rather than obviously the cryo-EM structure on the rise. But what's important here is to see that what's happening at very high resolution, notably something under 2 angstrom resolution. And here you can see that you have still also a quite large amount of structure solved with X-ray crystallography, rather than in cryo-EM, it's still kind of the beginning of the high resolution era. Of course, where there's no comparison is the size of the structure that are solved. Cryo-EM is much more interesting when it comes to larger complex. So X-ray crystallography is start to be shown more and more as a complementary technique, but it's still strong. And to illustrate this, we'll show a couple of projects which are very recently published. This one was published, I think, a couple of weeks ago. And it's a work from Nadine Renner in the group of Leo James. And it's a very interesting project, which I invite you to read. And basically, they show that the HIV capsid assembly is based on lysine ring around inositol phosphate ligand. So I won't tell you more because it's not my project, but I'm just inviting you to read this study. Another study, this time from Leo Kiss and David Noyers, which means there's probably NMR involved. I just read it. Yes, it's out yesterday, I think. So I didn't have time to read it thoroughly, but it looks very exciting, and with another complex structure at a resolution of 2.2. So I invite you also to read this very interesting study. Right, so that was just to show 2 examples of exciting study made by X-ray crystallography. Now, let's see what are these unfavorable properties of the crystal. When you look at this under a microscope, it's quite exciting because you got relatively good size already, and it looks symmetrical, there's sharp edges, so it's very exciting. So you may be so exciting that you grab your assistants, and then there's hope. You're kissing, it's extraordinary. You may buy a house in Cambridge with this successful project. Unfortunately, when you put it in the X-ray beam, it doesn't diffract, so that's very disappointing. But it's a scenario that can happen, unfortunately. So why this crystal didn't diffract? But we have to look at the internal qualities here of the crystals and how it holds together. And the molecules hold together with weak interactions, hydrogen bonds, and then the hydrophobic interactions. The contact area to make the crystal contacts are usually relatively limited. So it is something that is more flexible entity than anything else. It's not like a diamond. And because of the shape of the proteins, and also the type of intermolecular bonds, sorry, the solvent content is quite high, in the order of 50 to 70%, sometimes more. So that's why you may have some problems at the end with your diffraction experiments. And all these characteristics means that the crystal is not only seen as a single block, but more as a several little blocks that are not perfectly aligned, and this aspect is called mosaicity. The worst pathology of the crystal is the twinning. So now this time you have one, two or more crystals growing, similar crystals growing in different direction. So that's obviously doesn't help with diffraction or later with data processing. Now, before anything else, crystallization is to work on your sample. And the sample can be considered as the main variable. And I'm going to go through different aspects, not all of them, but just to say that you need to make use of nature, basically. So we'll usually try different expression systems that can make a radical difference in the behavior of your sample later. You will go through cloning, mutagenesis, and proteolysis. So these to alter the sequence of your sample, which more or less, depending on what you want to do and where you are in the optimization steps. But you will make use of databases, of course, to do sequence comparison and try to predict as much as possible certain things. Also, obviously, if it's a novel sample, it will be hard to predict anything about crystallization. But it's worth having a look of what was done before with similar ones. In the same way, we need to have a look at literature. So maybe someone in the past with similar sample find a stabilizing partner or stabilizing ligands, or try different buffer, some additives. So it's important to have a look at what's been done before. And then there's all biophysics assays, and there are a lot of them that could be helpful. I will just mention one, which is differential scanning fluorimetry technique. We'll see that in a few minutes. So there's also just an example to illustrate that a single mutation can trigger crystallization, and it's unfortunately the case for a protein of our lens in the eyes. And inherited congenital cataract is showing that it's just a single permutation that make the protein crystallize and create this pathology and pathology. That's just an example of what can trigger. But a single mutation usually is not, going to make a big change in your crystallizations, but it's something that will be used later during optimization step. Usually you will need more. You have a problem of the protein that wants to be crystallized. You have more problem that the protein wants to aggregate or polymerize. And polymerisation is being control of polymerisation is often a important work for some project, and notably the cell division proteins. And it's the case here with FTSQ, a protein that Fujinita from structural studies worked a lot before she was able to crystallize it. So we are talking about dozens of constructs here to finally be able to successfully crystallize this protein. And obviously, this is not a work only on construct, but as construct may be some work for optimization for purification, concentration. So it can become a lot of work. This part is very tedious sometimes, but it's necessary. A common technique used to help with that is the limited proteolysis. So what you do basically is a short and controlled proteolysis of your sample, and in the hope to remove the disordered chains outside at the surface of the protein. You will need mass spectrometry afterwards to deduce the new sequence that you generate, that you need to generate to produce this protein without these unfolded parts. At the LMB, we have a great facility for that. And Mark Scale and his team can help you with this kind of protocol, and there are many more that may help. What can help too is the NMR, and here I've got a slide kindly given to me by Jane Wagstaff from the LMB NMR facility, another great facility we have. And this technique is to try to find what part of the protein backbone is the most dynamics, and basically try to find flexible loops. Transverse relaxation. I'm not well versed on that, so this is just to mention that it's a tool that you can use to help to find a more crystallizable sample. Basically, here you see a plot with this transverse relaxation against sequence, and the higher the value of this T2 value is, the more dynamics you have. And in this example, it helped to successfully crystallize a sample after removing the corresponding flexible loop. So far, what we've seen is about reducing the size of a sequence of the sample and simplify it, rigifying it. Sorry, but not always the direction that is taken. Sometimes you produce a more complex and larger sample to be able to crystallize it, and that's the case often with GPCR studies. And you can see here the beta adrenergic receptor engineering. On the left, you have the GPCR is fused with T4 lysozyme, which crystallizes readily, and that helped to crystallize the overall structure. Another approach was to make a complex with the antibody fragment. So that makes the size of the sample quite bigger, but that was the only way to stabilize basically the GPCR and have it crystallize. In the same way, the group of crystates is using nanobodies to stabilize their GPCRs. And they're using other techniques, but you can ask specialists Pat and Tony what they do with that. They will know much more than I do. Just to mention one of the biophysics assays that can be used before crystallization to help to find a more stable sample, maybe it's a thermal shift assay. So basically you use a fluorophore, which fluorescence increase when it bounds hydrophobic core of the protein, which unravel when the temperature increases. So that's one way of screening, for example, some additives and see which ones stabilize your sample. So the limits now of all those alterations, well, obviously, at the end, you want your sample to stay meaningful in terms of the biological question to be answered. So it's nice to have crystal structure, but if your protein is far away from where it is in the cell, obviously it's a bit less interesting. And the limit of the preliminary investigations, like the previous assay we saw on the slide, and techniques like NMR, well, it's not always doable. It depends on the sample. Sometimes it works, sometimes it works less well. And also an important aspect in all of this is that so far we are working at relatively low concentrations of proteins and reagent tested. When crystallization, we're going to see that in a minute, it's happening in saturated conditions. And also, you are well in control with all those assays, but because you are investigating a single parameter, whereas during crystallization, there's a lot of things happening at the same time. Anyway, the bottom line is that the molecular weight is one important aspect. Of course, you want to try ambitiously high to have the most interesting structures possible, but you often have to try to have a lower molecular weight sample. It usually increases your chances. Purity, obviously, is an important aspect. And if anything else, another test, look at your SDS page, see if you see a purity good enough. Concentration, so 2 mg per ml for the sample is a minimum, but 10 mg per ml will be a better start. And the aspect of solubility, it's good to do simple tests early to dissolve the sample, because obviously you want to see more the effect of the salt that you're going to add during the crystallization. If your sample is extremely happy, too happy in its solutes, it may be against crystallization later. Simply add the effect to see the effect of adding adding glycerol as well in your sample. Maybe try 5–10% glycerol and see if it's making a difference. And also the buffer. Not bad idea to try different buffer pHs and see what happens with that too. Now, let's go to see some aspect of crystallization experiments, and the main one will be solubility. There are many parameters, but solubility is one start with. You've probably seen this type of phase diagram before. So only 2 parameters are shown here, the concentration of the protein and the concentration of the precipitating agent. So it's only 2 parameters, but it's a way to represent it. Here you see the limit of solubility shown with a zone where undersaturated and supersaturated. And in the supersaturated zone, you have 3 different zones. And you have the nucleation zone where we are quite highly saturated now environments, and that's where the protein usually will preferably nucleate. And the metastable zone is an interesting one because that's where the protein usually prefer to grow. So when you increase the concentration of your precipitating agent, most of the time you make a trigger precipitation. So you have a competition at some point between precipitation and nucleation, which is most in favor of precipitation, unfortunately. But that's what you can observe here. This type of phase diagram you may be able to do some for some small stable monomeric proteins, but in most of the case you won't be able to see such clear zones. And that's just a representation of this. So the experiment itself here, vapor diffusion. So basically you have a droplet formed with the sample and a more or less saturated conditions with the precipitant. It's in a sealed chambers, and the droplet is above the condition itself, which is much more concentrated in precipitant than the drop. And you have equilibration through vapor diffusion. And then in the drop, the concentration of the proteins will increase, the saturation is increasing, and that's vapor diffusion experiments will occur depending on the condition, the precipitant. It will happen within hours or within days. In the background, the precipitants have different mode of actions and different properties, but if you look at a simple properties, the salting out. So basically, with the increase of salt concentration, the hydration of the protein will decrease, and the hydrophobic interaction will be favored, and the sample will preferably bound to itself now rather than the water molecules. Although on the figure on the right, the bottom right figure, which represents precipitation, there's no water molecule around the sample. You still have some hydration though, even when the sample precipitates or crystallizes. Another important aspect is the pH. So obviously by varying the pH, you're varying the density of charges on the landscape of the protein. So that will directly affect the solubility. It may also give a different possibility of interactions between the macromolecules. So that's interesting for crystallization, of course. Another component of your condition will be an additive, so they may tune your the solubility, but they have a lot of other possible effects. Here, I like this one. This example is an interesting one because you have a crystal structure that were obtained only when nickel ions were present in the buffer, and that's because these metal ions were bound to the east side of the proteins and they triggered the formation of a trimer, which were not observed naturally in the cell, and only this trimer crystallized. So in that case, the additive was essential to obtain crystals. So this type of experiments now are happening in nanoliter droplets to save a precious amount of sample. And I started with saying that, yeah, the physiological protein-protein interactions are well characterized, but while the nature of the crystal packing contact varies in case-to-case basis, so you don't know what components to use to make it right. So it's all approached as a stochastic process, and we for this. Screens are composed with different components and formulated in different way. So if you want, for example, to see systematically what's happening against a pH and a concentration, you will formulate a grid screen. The problem initially is that you have so many suitable reagents that need to be tested that you will end up with millions of combinations. So there's alternative to reduce the number of conditions. One is the incompleted factorial, so which is to some extent a large incomplete grid screen where that you will formulate according to whatever reagent you selected, and also in a more or less random way to omit some combinations. Another alternative is to use the sparse matrix formulation. So this time you don't compose a screen with different reagents. You looked at conditions that were successful in the past and you put them all together. There are other alternatives where you place the additives are main component in your formulation, which is the concept of the silver bullet from Macpherson and Company. So these screens are usually found as kits of 48 conditions or 96. And just to show you a very traditional formulation here, the salt is what I like to call additives. It's not always a salt, but the component in lower concentration is called salt or additives. Most of the condition has a buffer, of course, to be controlled, more or less in control of pH. And the main component is can be considered as precipitant. So now you have a lot, usually this component in higher concentration. And you can see, for example, here 30% NPD at 2 molar ammonium sulfate. So the way we initial screen at the LMB is we supply pre-filled crystallization plates. So the kits in tubes are aliquoted into crystallization plates, which are stored and made really available to the user, which is good because you don't never know when the sample will be ready to be used, freshly prepared. So all the screens are available anytime there. This slide was an opportunity to thank Steve Scotcher and his group at the workshop, which are regularly making spare parts for us or customize our equipment. Our main strategy is the large initial screen. So we actually have 22 pre-filled plates called LMB plates. So they have a lot of all different screens. The advantage is that you can have, you should go for this large initial screen or select, cherry pick a different type of screen depending on your sample. And we use the mosquito to do a Vapour diffusion droplet of 100 nanometer protein plus 100 nanometer conditions. So what are the pros and cons of our main strategy? Well, the advantage is that you, as you saw in the slide with the poor yield of usual crystals, you may need that many conditions to find an exclusive hit. Because of this very low yield. If you have more hits, then great, because you can investigate crystal that grow in different type of conditions. So I don't think you never have too much hits. Well, for sure, you reduce your requirements of optimization. So this may mean that you have less work on sample preparation or crystal optimization later by screening more initially. Disadvantages, obviously 22 plates, that means 352 microliter required overall to set up the drops. Could do with a bit less with using some tips. But that's the order of amount, the volume we need. And there's a lot of plates to look at. It can be a bit tedious to assess all those experiments. But you know, if you want to take a real chance with a novel sample, it's worth to do a large initial screen. So the assessment of the experiments, actually, well, fortunately, you will regularly see precipitates of crystals. As a rule of thumb, 50% of precipitates across all your plates is probably in the right zone. Also, it may mean something, it may not. It may mean that your protein is very unstable if you have even more precipitates. But that's kind of where you want to be. Sometimes you see things in your drop, you're not sure if it's microcrystals, but you see sharp edges. So it's difficult to know when you see these kind of things. UV light may help. Also, if your sample is poor in tryptophan, you may not see any signal. There are other occasions as well where you won't see the signal out of UV signal out of a crystal, but you can try that. Here it's a more convincing results where you actually see the phase diagram in a droplet with precipitant, microcrystals, and crystals. You can be almost sure that here is protein crystallization, and it was in this case for a project of Mark there, who nicely provided this picture. The only way really to check is to shoot your crystals and see if it diffracts. You know, for me there's no other way really to analyze the data, because that's what you want at the end. You don't want nice crystals, you want nice diffraction patterns that will enable to solve the structure. And Dom Bellini at the crystallization, X-ray facility is responsible for a powerful X-ray source that we have in-house, which enable us to pre-screen the crystals before shipping a batch of crystal to the synchrotron. So that's quite good. Also, to analyze the formulation of all the different heats you may have, we developed a freely available software, a web-based interface where you will select your heats and you can produce the formulation of different conditions under an Excel file, which can help as well. Time is running out. So quickly, the optimization protocol, it works to put some efforts on optimizing your conditions to have bigger crystals. So this can be done in different ways. For sure, you must get in control of your of the conditions you're working with your own stock solution. And for this, we start always by the four-corner method, where you basically make manually at a relatively large scale of few milliliters, the condition corresponding to 4 corners of the plate and then the robot will use this 4-corner solution to produce a linear gradient of 2 of the components, 2 of the reagents. So that's really the step you will start with for all the conditions of interest. Once you're in control of your condition, then you can play a bit more with it. And one straightforward way is the additive screening. And before we were using additive screening deep well block, transfer in plates. As for the initial screen at the LMB, we have MRC plates with pre-filled with the additive screen and that are keep frozen. So that's facilitate as well the additive screen. So no excuses not to try that. It's very straightforward. So you would basically add the condition that you prepare yourself now based on the result of your 4-corner solution protocols, and you will add it to the plates with the additives, and then set up your plate normally. Seeding may be required. Seeding works really well. And obviously, an ugly side, I just have to read it, but I wanted to make sure say it right. The fact that crystal growth professionally occurs at lower saturation than nucleation means that a technique like vapor diffusion can be limiting because nucleation competes with growth, while saturation keeps increasing. If you remember, on the vapor diffusion, you will have all the concentration will increase until equilibration, of course. But you may have the crystals nucleating early, and then you're still going toward the phase of higher saturation, which is not ideal for growth. So a way to decouple nucleation and growth is to take microcrystals, which will be the seeds, from your previous trials, and to use them to initiate crystal growth at a lower level of saturation. So we now transfer your initial microcrystal that occurred in some experiments and transfer them in other plates, in other drops, where you have a lower level, you start with a lower level of saturation, and hopefully this will optimize the growth of your crystals. A modern approach to this, which may be easier, is to basically now you're going to take any kind of crystals that you produce initially, and you put them in an Eppendorf with a kind of bead, and when you vortex it, this is mashing up all the crystals. So they will have a lot of different size and shape and type of crystals that you get, and you will rescreen, like proceed with an initial screen again. So I say rescreen because now you're not trying to be in control with the same type of condition, but you reintroduce different components in your experiments. This technique is quite successful as well. And there was the phase diagram here showing what's happening. I won't have time to go through that, but basically the idea is the same as classic seeding. You want to work out a growth on the metastable zones, lower saturation. And with this random microseed matrix screening, you also integrate new reagents at the same time. So it's a bit like an additive screening and seeding gathered in a way. So just few tips, further tips if you fail to optimize your sample, your crystal, sorry. you can try to heat shock the sample. So that basically the idea is to get rid of a sample that will be a bit denatured before you proceed. That's a cool trick. Of course, there's a scale up, so you can also try to prepare the drop initially, so that will change the parameter of equilibration during vapor diffusion. And you could try it if you have enough sample, of course, to do that initially. Mixing the droplets. When we proceed, we don't mix the droplet, the conditions and the protein outputs together in the protein well of the plates, but they're not mixing. This is something we were doing before, more with the manual pipetting. But you can do it on the robot once the drops are big enough. There's a protocol that enables you to mix the drops. So there's something to try as well. That may be able for you to be more in control. If you remember the slide with the phase diagram droplets, where you see precipitate and microcrystal, that could be a sign that you had diffusion in the drops before the vapor diffusion or during the vapor diffusion. So this is nice because it triggered crystals, but that means that also it will be harder to be in control there. And the way to be more in control is maybe to mix your droplets first, but you don't know if it's a good idea or not initially. Different ratio is definitely something to try. So now you will not put 100 nanoliter of protein and 100 nanoliter of conditions. You will use different ratio of both, again, to change some parameters of the equilibration. This different source of reagent, that's one of the reasons why also we use different strains from different suppliers. It's a source of reproducibility issues. Are you really in control with your buffer? Are you really in control with your reagents? Polyethylene glycols are very unstable, often source of reproducibility issues. So you need to think about that. And if you really cornered with reproducibility issues, well, you can simply pipette out the condition from the very initial plate that gives crystals that could never be reproduced, and use these conditions as an additive to bias the follow-up experiments. I'm reaching the end of my talk. I will just mention some tutorial and videos that is good to look at. And notably, if you go on the site of Terence Bergford's, you will have a lot of material there, especially for seeding protocols, but other things. It's very useful. I would also advise to go to see a publication of Yorgo, what is from the university wing here, that he made a few years ago. It's a very interesting starting point with a video showing the characteristic and the setup of vapor diffusion and all the techniques for crystallization. And we have on the LMB crystallography courses page from 2013. It's not recent, but what's good is still good now. What was good then is still good now. And it's been a very interesting presentation from Mendert Leimers, who is now left us for Leiden University Medical Center. So I advise you to have a look at those videos. Books, again, Therese Beckford's books specifically on protein crystallization. It's very nice. Alexander McPherson book on macromolecular crystallography. I really like it. It's very clear, and you will understand a lot through with this book. Also, like other books, especially the one from Bernard Rupp, a very extensive book about all the macromolecular crystallography. And this last book, Crystals, X-ray proteins, more on the theoretical side, I think, but all interesting. And I will thank the group of Jan Lore from who are studying the bacterial cytoskeleton, and they are still doing a lot of X-ray crystallography, and they work hard because I can't, you know, all the data, most of the data I have and I've shown here is the result of the hard work of the people from this type of group or the group of Roger Williams as well. That's why I really thank them. I also thank the group of David Barford, which are working on chromosome aggregate segregation. And we are all together now under the big family of the UKRI. So I think this is it. And if you have any question, you can contact me by email. And just to mention that this talk is based on the latest article I published, which is called A Beginner's Guide to Macromolecular Crystallization. It's in the Biochemist. I forgot to put it there. All right, time is almost up, so I don't disturb on the Q&A bits. Oh, there is a question showing up, actually. Is there a method to improve diffraction pattern from a crystal aside from growing better crystal? Oh, well, that's a good one. Aside from growing better, yeah. So you mean, yeah, outside from crystal, yes, because you can spend a lot of time. I see what you mean. Trying to optimize crystal conditions. On the top of my head, there are some methods. For example, annealing. So you will block the cryostream that is between the crystals. That is keep the crystal frozen for a very momentous time and see what happens there if you have a slight dehydration of the whole experiments, and that may kind of improve your diffraction. But growing better crystal, I'm afraid, is still the most the main aspect behind this. Everything else will be about working with the crystals, again, like this annealing methods, or try different cryos as well, because obviously when you are cryoprotecting your crystals, you have this differential expansion between the solvent and the crystals, and that's why it's a good idea to try different cryos. There's some oil as well that you can try instead of cryo. This is something that maybe Dom Bellini will talk about in his talk on Thursday, in 2 days' time. Is there another question?
Yes, you have one question in the Q&A. If you open the Q&A box.
I did, but okay, sorry. What is your typical sample concentration for initial screening? Ah, that's a good question. There's no typical concentration for sample screening. Personally, I will try to concentrate the sample as much as possible. So for that, I will sacrifice a small part of the sample that I have eluted and see how far I can go until it crashes by doing the concentration steps, whatever method you are using for that. And so now I know my limits, my concentration limit of my sample, and I will concentrate the rest just below that. That's how I proceed with a novel sample. Or I was anyway. I'm not doing this that much anymore. But if I have a novel sample that I had to work with in the future, I will still proceed this way, because I prefer to have it too concentrated, see that it's too concentrated afterwards, because then I can simply dilute it and redo an initial screen with a lower dilution, because if it's not concentrated enough and I see only clear drops, then I have nothing. I have no indication whatsoever. Actually, having of precipitates in the in your drops like a high proportion of precipitate. It's not such a bad thing because the small proportion of the clear drop are becoming very valuable information. So you could use the formulation of the conditions to actually try to optimize your sample during its preparation now. You have very valuable information about what's happening at higher saturation. Now, if you don't want to take this extreme approach, so to speak, you need few mgs minimum. That's a start. And I will say, as a rule of thumb, 10 mgs is a better start. But personally, I like to start with concentration sample as concentrated as possible, and also not only concentration wise, but you need to work on the desalting your sample, which usually will decrease the possibility of going higher in concentration. But this is another aspect that you need to work on as much as how concentrated it is. All of this is sample optimization, basically. But thank you for joining, and that was a good question as well. So thanks for that, and I hope to see you at the LMB Crystallization Facility to trial your sample for crystallization. Goodbye.